Great. Okay, we can start.
Thank you. Thank you, everyone. So hello, everyone, and welcome. And we are very excited and happy to have you join our fourth MoroccoAI webinar of this 2022 AI webinar series. A brief intro about MoroccoAI and who we are. We are an initiative led by AI experts in Morocco and abroad. These experts are professors, researchers, and AI professionals. Our main mission is to promote AI growth in Morocco and build a strong collaborative AI community, both locally and abroad. We have launched different activities such as AI webinar series. The AI webinar series aim to bring AI researchers to present and share their work with the community. For today's webinar, Redouane and I will be your hosts. I will let Redouane introduce himself.
Hello, everyone, and welcome. My name is Redouane Gansat. I'm a research engineer at the IPSL Climate Modeling Center in Paris, and I'm very happy to join Iman and join you all for this webinar. Very looking forward to Dr. Daouda's talk, and welcome again.
Thank you, Redouane. And I am Iman Khouja. I am a data scientist, and I have a PhD in NLP. And don't forget to follow us on LinkedIn, Twitter, and Facebook, and join our community on Slack to be notified about our upcoming webinar. In this fourth webinar of 2022 webinar series, Dr. Tariq will be presenting AI for immunology, where he will be introducing AI works on single-cell representation, zoonotic transition, and prediction of markers of infection. Dr. Tariq Daouda specializes in applications at the intersection of artificial intelligence and immunology. He holds a bachelor in mathematics and computer science from Université de Lorraine, a master's in machine learning, and a PhD in bioinformatics from Université de Montréal. Then he pursued a postdoctoral training where he was affiliated with the Broad Institute, Harvard Medical School, and the Massachusetts General Hospital. He is now a research and education fellow at the University Mohammed VI Polytechnic. His research focuses on the development of novel artificial intelligence methods to help answer questions about the human system. Over the years, he has worked on identifying markers of cancer and other infections and analyzing viral sequences to identify strains likely to transition between species. If you have any questions during the presentation, please type them on the chat on Zoom or YouTube, and we will make sure to bring them up at the end of the presentation. And please be sure to stick around for our networking session right after the presentation, where we will break out in smaller rooms, so you get to meet and talk with some of the attendees. And the presentation will last for approximately 30 minutes, followed by 10 minutes Q&As. And without further ado, Dr. Tariq, please, the floor is yours.
Thank you, Iman. Thank you. Thank you, Iman. Thank you, Khalil. Can everybody hear me?
Yes, we can hear you.
Thank you. So I think it's time for me to share my screen. Okay, is everything all right? Can you all see my screen?
Yes.
Perfect.
Superb.
Thank you, first of all, MoroccoAI, for the invitation. I'm really thrilled to be able to present my work to this audience, and I feel very happy, essentially very happy to be able to present to MoroccoAI. So I start with this very general slide about how I see AI. So I see AI as a telescope for the mind. So basically, as the same way as a telescope allows us to see further and things we wouldn't be able to see with the naked eye, AI can allow us to do basically the same thing for data. So instead of taking this big data, this huge amount of data for which our brain is not designed our brain is not designed to process this kind of data, we can pre-digest it through AI algorithm, and then in a way that allows us to accelerate research, to basically understand what's happening. So this AI digestion is basically what I specialize in, and this is what I'm going to present in the following presentation. So the presentation is entitled Artificial Intelligence for Immunology. And the first work that I'm going to present is about understanding the origin of cell markers. So more specifically here, I'm talking about MHC1 associated peptides. So MHC1 associated peptides are basically small molecules presented at almost the surface of all our cells. So our cells produce proteins, and then these proteins will be cut into small pieces, and these small pieces will be presented at the surface, and the immune system will then check them to see if the cell is behaving normally. Because if a cell gets infected by a virus or becomes cancerous, then it will present abnormal peptide because it will produce abnormal proteins. And when the immune system detects these abnormal peptides, it basically kills the cell. So this is a cornerstone of the adaptive immune system. And what's really important is that if we understand it better, we can basically understand better cancer, viral infection, autoimmune diseases, a lot of things that are linked to the adaptive immune system. So where do these maps come from? So they come from proteins, as I said. So the proteins get cut into small pieces, presented at the surface of the cell. But the proteins themselves, they come from mRNA. So mRNA is translated into proteins, and I'll get into that into more details in the subsequent slides. But the idea is that mRNA becomes protein through translation, and then these proteins get cut into small pieces, and then these pieces get presented at the surface of the cell. So we already have predictors for the protein side. So we can run predictors on proteins to see basically if these peptides would fit into these molecules that are called MHC1 molecules that present the peptides. And the problem is if we just do that, if we take an MHC1 prediction affinity and run it on the proteins, we get a very high false positive rate. And by very high, I mean 80 to 90% false positive rate. So that tells us that there must be another explanation. The binding affinity of the peptide to the MHC1 molecules is not enough to explain their presentation. So there's probably something else that we haven't thought of. And this thing probably, maybe it comes from the mRNA. So that was our first hypothesis. Because mRNA translation is actually really hard. So you have a big machinery called the ribosome that goes and reads the mRNA 3 by 3 and then translates it into a protein. And the huge complexity comes from the fact that only 21 amino acids for 64 codons, and then over 600 tRNAs. So every codon is translated into an amino acid, and there's basically 30 times more ways of translating codons to amino acids than there are amino acids if you look into more into the tRNAs. So from a more machine learning perspective, more of an NLP perspective, we can consider the mRNA or DNA is basically the same thing here. So I'm sorry if there are some biologists around, but for this application, it's basically the same thing. So you take the mRNA is basically the sentence, and then you have words inside the sentence that are basically 3-letter characters. So you probably know that the DNA only has four characters, so it's A, T, G, or C. If you put them into blocks of 3, they become what we call codons, and these codons are basically the words. So or the tokens, if you want to speak in an NLP fashion. And these tokens get then translated into another set of tokens that are amino acids, and a protein is just a string of amino acids basically. So you have 64 of these codon tokens and only 21 of these amino acids. So you must have a redundancy. So some of them are synonymous, which means that they translate into the same amino acid. But what we're showing here, what I'm going to show actually in this work, is that they're not that synonymous. Even though they translate to the same amino acid, their effect is actually different when it comes to the presentation of these MHC1 peptides. So we are going to build a map predictor. So this is the central dogma of biology. So you have mRNA translated into proteins, and then you have these proteins cut into small peptides, and then, for example, this green part gets translated into this green protein part, and then this becomes a peptide. And then the goal is to say whether or not these green peptide will be presented at the surface of the cell. Will it be a map or not a map, basically. And the way we're going to do that is by taking only the flanking region. So you have, this is the mRNA or the protein sequence, and then you have the part here that codes for the peptide that's presented at the surface of the cell. And the goal here is to say, if we just take the flanking region, and the rationale behind that is that the ribosome actually reads the mRNA sequentially. So if we take only the flanking region, will we be able to say whether that green part is a map or not a map? And we'll do that through a neural network, and we use NLP techniques. So the same standard NLP techniques that are used now. We tokenize, basically transform to codons or amino acid, and we embed, and then we try to predict whether whether the sequence in green in the middle that the network never sees will be presented as a map at the surface of the cell or not. So we actually built two networks. So the first network takes mRNA sequences, and the second network takes protein sequences. So we have a total of approximately 120,000 sequences, mRNA and protein. They're basically the protein are basically the mRNA translated. So, and we're going to put these two network one against the other, because what we're trying to see here is whether the mRNA holds more information when it comes to the presentation of these amino-associated peptides than the protein sequence. And if we run this experiment, it can only go into two different ways. So the first one is the mRNA predictor is better. That means that the mRNA holds information with regard to the presentation of amino-associated peptides that is lost during the translation of the mRNA to the protein. Or we get exactly the same thing. The mRNA predictor and the protein predictor give basically the same results. And that means that there's no more information with regards to the presentation of map sequences. And again, this is possible only because we have 64 codons that translate to 21 amino acids, which means that there are some codons that are synonymous. So that's the reason why this experiment can only go these two ways. So, and these are the results we got. So this is on the x-axis, you have the context size. Basically, the context is the size of the flanking region that I've shown you in a previous slide. And on the y-axis, you have the AUC representing the accuracy of the models. In green, you have the amino acid sequences, and in blue, you have the original mRNA sequences. And what you can see here is that for every context size, basically, it doesn't matter how much the size of the flanking region that you take, the mRNA network always has a higher accuracy than the amino acid networks. And what that tells us is that the mRNA actually holds information when it comes to the presentation of these amino acid peptides, these maps, that is lost in translation when we translate these sequences into proteins. So this is basically the genesis of the network that we're going to use afterwards. It's called CAMAP, Codon Arrangement Amino Acid Peptide Predictor. And what this shows us is something that wasn't known before in the literature, because everybody was focusing on proteins. So for the first time, we have this very clear demonstration that actually there's something in the mRNA that's very important to the presentation of the amino acid peptides. And we went even further. So the nice thing about neural network is that you can give them any type of data and they can analyze it. So you can build predictor basically on any type of data, which you cannot do with a human brain. That's very hard. We're very good at, for example, image analysis, but not so much as to do DNA analysis. But the neural network basically doesn't care. And the nice thing is that if you can build that predictor and then you can analyze, interpret that predictor, then you can derive some meaningful biological insights. And this is what we did here for that project. So what you see here is the position in the sequence. So this is a context. We took a context of 54 codons on each side. And so these are codons on amino acids because we, as I showed you in the previous slide, they're better predictor than the amino acids. So here you have a codon, 54 codons on each side for the context. The blank space here is the part that the network doesn't see, and the part that for which the network is trying to predict whether it's going to be presented at the surface of the cell or not. And so, as again, on the x-axis you have the position, and on the y-axis you have what we call here the preference. So the preference is basically normalized probability, where you have 0.5 is basically neutral, and then 1 is the highest value, and then -1 would be the lowest value. And what the preference tells you is basically how codon affects the prediction of the network regarding that blank space right here. So what we see here is that if we take this amino acid serine, and there are 6 codons that code for serine, so 6 codons that were considered to be 100% synonymous, what we see here is that they have basically the same effect. There's some differences, but basically what this tells us is that if you have a serine right before the sequence right here, what it tells you is that there is, it increases basically the probability that this sequence will be classified as a map sequence versus a not map sequence. And so we have basically the same thing here for proline, but in reverse. So you have proline is another amino acid that has that can be coded by 4 codons. So you can see that the major effect is again around the sequence, the map sequence right here. And you can see that having a proline, it doesn't matter which codon for the proline, decreases the probability that the sequence will be classified as a map. However, things become really interesting if you look at different amino acids. For example, if we take here tyrosine, tyrosine is only encoded by 2 amino acids, 2 codons. So you have TAC and TAT. So both of these codons, they are translated into the tyrosine amino acid. And what we can see is that although they're translated to the same amino acid, they have opposite effects on the prediction. So what the network is telling us here is that if you have a TAT right before the sequence, then the sequence is more likely to be a map. However, if you have a TAC, then it is less likely to be a map. And this is very interesting because as of now, these 2 codons were considered to be 100% synonymous, but really they're not. They have opposite effect when it comes to MHC1 acid peptide presentation according to the network. Here, if we look at another amino acid, leucine here. So leucine is encoded by 6, can be coded by 6 different codons, and we have even more diversity here. When we look at the leucine codons, we see that 2 of them are basically neutral, so they don't change the prediction of the network. However, one of them, CTA, increases it, while the other ones, CTT, TTA, and TTG, decrease the prediction. So what this suggests is that although these codons code for the same amino acid, they actually can have very, very different effect when it comes to the presentation of the MHC1 acid peptides. So this is very nice, and this is something that I'll come back later during the presentation because we moved forward with this data. But now I'm going to show you something that's also very interesting because it's nice to be able to build a predictor. But if we trying, for example, to build a predictor for a new vaccine, for example, a cancer vaccine or virus vaccine, does this CAMAP that we've just introduced improve the prediction compared to what we had before? And this is what we answer here with these 2 figures. So we knew some things before. So we knew that ligand score, basically the affinity of the peptides to the MHC1 alleles, is very important. And we also knew that the expression of the mRNA is very important. So these were the 2 major predictors that we had before. And when we add CAMAP, does it increases the accuracy of the model or not? And this is what we find here. And it increases it significantly. So this is very interesting. So it means that with CAMAP, we actually found new mechanisms, new biological mechanisms that explain the presentation of MHC1 peptides that were not known before. So here, this is what you have with the ligand score alone. This is what you have with the expression and the ligand score, and this is what you have with CAMAP added to the expression and the ligand score. So you get a way better accuracy here, significantly better accuracy. We also ran a very bold experiment. So we trained here a network on human cells. So this is the data set we trained the network on, human BLCL. So it's a type of blood immune cells. And then we applied that same network without retraining it. We applied it to different data sets. So first of all, we applied it to other types of cells, of immune cells. And we can see that the network is still able to give accurate predictions, even though it was not trained on these cell types. And then we took exactly that same network and applied to mice cells. And this is something that's usually not done, because you basically train on a specific data type, and then you apply the network on usually the same data type. But in this time we applied it to another species and other cell types into that species, and we can see that we still get accurate predictions. And this is very interesting, because what it shows is that the features that CAMAP learned are actually conserved across species and cell types, and that means that they're probably very, very important. Also, when regards to prediction, we have different kinds. We can create different tiers, basically, of Artificial Intelligence peptides. You have the best therapeutic targets, and then you have those that are not the best therapeutic targets. And the best therapeutic targets are the ones that are basically the 1% hit, the highest, basically the one that you would look for if you're trying to make a vaccine. And what we see here with CAMAP, and this is very interesting, is that the result when we add CAMAP to the ligand predictor and the expression, what we see is that we dramatically decrease the false positive rate when it comes to predicting the 1% top hit. So we go from 32% here to only 1%. This decreases as we increase the size, basically, of the hit. So 5% to 10%. You can see that the impact of CAMAP decreases. However, for the top therapeutic target, is very striking. We really go from 33% false positive rate to only 1% false positive rate. So if you recall the first slide that I showed you, when I was talking about the high false positive rate, I was basically talking about this number right here of 72%. So we are able to go from 72% to 33% when you add the expression, to only 1% when we add CAMAP, and this is very encouraging results. So, and then we did something that in my opinion is very, very interesting, and I would like to see more of this kind of research done in the lab. Is that we took the CAMAP network that we've trained, and then we asked the question: is it really biologically relevant? So we have these features that the network has learned, but are they really what we think they are? So we designed an experiment for that. That experiment is very simple. So we took the CAMAP features and we took a protein sequence, and we modified the protein sequence without changing the amino acid sequence, just the codon sequence. So the translation, basically the codons translate to exactly the same protein. So we just changed the codons, the synonymous codons, according to what the network told us. And doing that, we generated two different proteins. So we had the wild type protein right here, which is our baseline, so the protein in its normal state, and then we designed two different proteins from that sequence. One of them is the enhanced presentation, and the other one is the reduced presentation. So the enhanced presentation is predicted by the network to present a peptide more, and the other one is predicted by the network to produce that peptide less. And what we see here is that the network actually made accurate predictions. So the sequence that we've designed according to the features learned by the network are actually biologically relevant. So the network was actually right in his predictions, and this is very interesting to be able to confirm the results obtained through machine learning research into that fashion. We also ran another experiment when it comes to the translation, and I'm not going into details for that experiment. If you have any questions about that, feel free to ask it during the question period. So this is super interesting because we have created a way to basically design AI-informed sequence engineering, and this is, in my opinion, very, very interesting and very exciting work. Then we also have tried it for viral applications. So we tried it on SARS-CoV-1 and SARS-CoV-2 epitopes. Epitopes are basically maps. We tried it on the prediction of SARS-CoV-1 and SARS-CoV-2 emission peptides. And what we show here is that CAMAP is able to accurately predict these maps for the two SARS-CoV viruses. Very, very accurately. So you can see like the prediction is relatively high, and also better than the other methods. So I can go into more details for that part also if you're more interested in during the question period. And using that method, we were able to basically design a cocktail of peptides that would cover 78% of the US population and over 95% of the virus strains for SARS-CoV-2. So this is also a very exciting application of CAMAP, and I'll be very happy to go into more details in the question period. Okay. Now I'm moving to another project where we actually learn from mistakes made by the network. So this is a project that I'm doing with the students of mine that I have the pleasure to co-supervise. This one had to be from the NZS. Okay. So we know that viruses can jump from one species to another. So we have, and in this case, we're more interested into the influenza virus. So influenza virus can change hosts from swine to human to avian. So this is something that has been documented before. And the theory says that basically the virus does that through mutation. So the virus mutates, becomes more adapted to a new kind of host, and then can jump from one host to another. So for example, this would be a sequence for a human influenza virus. You have a mutation here, makes it more likely to go to infect avians. You have this mutation here, makes it more likely to infect swine. And the goal of this project is to basically be able to identify these mutations that would increase the probability of the virus jumping from one species to another. So we did that all again with using NLP models, and this time we use LSTMs and transformers. So we took 2,000 sequences, mRNA from mRNA influenza sequences, and these sequences are associated to either human, avian, or swine. So we got very high accuracy, but in this case, what you're more interested in is the mistake. So if you have 98% accuracy when it comes to predicting which host is associated to which sequence, what can we learn from the 2% mistakes? And the theory here is that if the network with this very high accuracy makes any kind of mistake, it's probably because the sequence that has been misclassified bears features that are not common for that host, and that are maybe more representative of another host. So this is what we're going to endeavor and try to do, is to find these markers. So we can have a quick look at what our predictions look like. So this is a UMAP reduction of the latent space. So you have here the three hosts. You have avian, human, and swine. And this is for the bidirectional SEM. This is for the transformer. And you can see that every dot here is a sequence. And you can see that the accuracy is actually very, very high. So you can see, you see this, like, sequences are really well grouped together, because it's usually easy for the network to differentiate which host corresponds to which sequence. And however, there are still some mistakes, and these are the ones you see on the right side here of the figure. And you can see that these mistakes are usually at the boundaries between different hosts. And these are the dark blue. The dark blue dots that you see here, they are all misclassified sequences at the frontier between different hosts. So what we will do here is we extract this misclassified sequences. We basically will ask the question, why were they misclassified? And we'll do that using the line algorithm, some statistics, and also some information theory to try to extract these reasons why they were misclassified, and from that derive the transition markers. And so this is what we did, and so you can see here that we took codons because our results also once again show that codons are better predictor when it comes to zoonotic transition than amino acids. And these results, actually, these Venn diagrams represent what we did find are the most important avian codons, and we put them together with results, with previously known results from the literature. And what's really interesting here is that these other papers here, they use biochemistry. So they're really biological methods. So our method is the only one that uses machine learning. And we can see that we do have a very interesting overlap between these other methods. So we are still able to predict the codons that are associated to avians that these methods were able to predict. And this is what you have here at the center of the Venn diagram. We did the same thing here, but this is for the only for the HA and NA gene of the influenza virus. So these genes are very important because they allow the virus to enter the cell and then exit the cell. So they are very, very important to influenza. And we can see the same thing right here. We still have a very interesting overlap between previously known results and what we find with our method. And if we combine all together, we get these three codons that are very interesting, very interesting candidates for transition from either swine or human to avians. And using the same method, we're also able to have candidate positions. So this is, for example, what we have for the codon AGA for the transition for a human to swine. So this is still very experimental. This is something new to my knowledge. I don't think anybody has the same type of analysis the same way we did it, but we are able to use the same methods that we use for identifying the codons and apply them to specific regions in the sequence. And this is what we find. So there's no, for example, at the moment we have no parallel with the literature because we couldn't find any that fit our purpose as of now, but we're still looking. But it's very interesting because it allows us to say, for example, if you have right here, if you have this codon AGA for the PA gene between position 651 to 681, that actually means that this strain is likely to move and to change hosts. So this is also very interesting work, and I'm very, very happy of what we did with this. So now moving to another type of work that I'm doing with collaborators, and this is about better immune cell representations. And this is more, this is about single cell sequencing technologies. So single cell sequencing technologies have really taken the biological world by storm in the recent years. Because before that, we were able to only sequence in bulk. So you take a tissue and basically you blend it and you sequence all that at the same time. And what that gives you is basically an average of all the cells you have. But now we're able to sequence the cells one by one. So we can take the immune cells, and every single immune cell, we can sequence its genome, we can sequence its transcriptome, we can sequence its proteome, we can get a lot of different measurements from single cells. And that's really interesting. But the issue is that we have all these different sequencing methods, and they all have their different biases, and they all have their different susceptibility to noises. And what we're trying to do here is to find ways to better represent all these methods and to integrate basically all these different measurements together into a more accurate vision image of the cell. So the first model here that I'm going to introduce is called fiber autoencoder that we've introduced a few years ago. And the fiber autoencoders are very interesting in the way they represent the latent space. So they have a very strong constraint inside them that actually separate the latent space into two different parts. So you have the base part here, and then you have the fiber space on top of it. So if you have, for example, conditions and variability inside these conditions, these networks have been basically designed for this kind of applications. So they're very interesting from a biological perspective because they allow us to represent, for example, different patients on the base, and then the variability of the cells of that patient on the fiber space. And so we can have, for example, different patients. We can have sick to healthy. We can have all different, or any type of condition that you might think of could be represented this way. And then once we have trained the network and the network has this latent space trained and learned, we can trace links. We can link basically the fibers together, and through geodesic transport, we can take a point, for example, that presents a cell in a healthy state, and then ask the question, what would that cell look like if it were sick? And this is why these models are so interesting at the moment, is that they really allow us to play with the different conditions in a very nimble way. And so this is what the fiber autoencoder model looks like. So it's here you have the condition, here you have the data, and then you can see that the condition basically becomes the base space, and the data, the image here, becomes basically the fiber space. And the way we do that is by having here gradient reversal classifier here that basically predicts the condition and tries to remove the information of the condition from the data. So if the network is successful in doing that, what you end up here is a fiber space that's basically orthogonal to the base space. So it doesn't contain any information from the condition. And then these two are then grouped together, they're basically concatenated and given to a decoder layer, because fiber autoencoders are autoencoders. And we try to reconstruct the image, the input here, from these two entries, base and fiber. And then we also have additional codes that you can add that are not necessary for it to work, but that can help increase the quality of the results. So we have a GAN cost here and another GAN cost here that actually ensures that the information from the condition is not lost. But these are really optional. The real network is this whole thing with the GRL here. That's very, very important. These networks are also, they have very interesting features. One of them is that they seem to be able to learn from very little data. And so this is what the network does when trained on a very small data set. So Olivetti data set only has 40 different individuals, only 10 images for each one. It's a very old data set. And we trained it on this Olivetti data set. You can see here the interpolation that we can get with only 10 images for every subject. And you can see here what we can do with this type of applications. For example, you can see what you see on the left side is the interpolation, and on the right side is the closest image from the training set. So training set only has 10 different 10 training example, 10 different images, but we're still able to generate that that smooth translation and 3D rotation for that person, even though we only had 10 images. So we can generate easily 10 times more images that we had in the training set. We can also do interpolations between two different phases here, again, with only 10 examples for each one of these individuals. And this is very interesting because it's for biological applications, because it's not rare to have rare cell types that you need to represent accurately. They also seem to be very noise resistant. So this is a very crude experiment, but still a very telling one. So what we did here is simply generate, this is very synthetic. So we generate five Gaussians, isotropic Gaussians in 128 dimensions, and then put randomly, just drop out 99% of the values, put zeros in there. So there are 99% corrupted, and you can see what happens if you do a PCA after that. There's the information about the fact that there were 5 Gaussians is completely lost. However, if you train a fiber autoencoder on this corrupted data set, you can retrieve that information. So you can see that some of it is lost here. Because the variance of the Gaussians is wrong. It's not, they used to have like the same variance on every dimension, but you can see that the variance here is skewed a little bit. However, the important information that these are 5 different data sets, 5 different classes is recovered, even though the data was 99% corrupted. Yeah, so we tried these networks on the single cell batch integration problem. So batch integration simply means that if you run an experiment, 2 different people in an experiment, you get basically two different results, even though they're the same cells, even though the machine is the same, etc. You still get some technical noise, and here we're trying to basically remove that technical noise. So this is what we call the batch integration, the batch removal problem. So we trained FAEs here, and on these are blood cells, and you can see that this is the data set as is. So you have two different different batches, the blue one and the orange one, and they're really separate. And then we trained the fiber autoencoder and asked basically the fiber autoencoder to integrate these 2 batches. And we do that through the geodesic translation transport that I've shown in the previous slide. And we compare that to another model, a version model called SCGen. And this is what SCGen does. So you can see that SCGen does it pretty well. So the integration is very well. You can see that the blue and the orange are really well mixed. However, there's one issue here, is that this little island of purple has been separated from the red. And these are monocytes. Both of them are basically the same cell type. They're not exactly the same monocytes, but they're close enough that they should be together, and they have been separated. So this tells us that some information has been lost. However, when we do the integration with the fiber autoencoder, they are still here. They're really, we can see that the monocytes are still here together, and you can see that the overall shape looks more like the shape we had before the integration, even though the 2 batches have been very well integrated. We run the same experiment here. These are pancreatic cells, and we try to integrate them. Every color is a batch, and we try to integrate them using SCGen first, and then the fiber autoencoder. And you can see that the SCGen here also does a very good job. However, it was not able to integrate these small red cells here. That's probably because the data set is very small. However, fiber autoencoder were able to integrate even the smallest batch.
So Tariq, could you please wrap up in 5 minutes?
Sorry, I couldn't hear you.
Could you please wrap up in 5 minutes to leave time for the questions?
Okay, so you want me to stop? I can't hear you very well.
To wrap up in 5 minutes, if you can.
Yes, that's okay. Don't worry.
Thank you. Thank you.
Yeah. So the next thing we're doing here is doing the same thing, but we're trying to integrate different batches together, but also integrate in different modalities. So we have mRNA and protein sequencing for the same cells, and we try to integrate them into a single representation. And you can see here that the mRNA alone, the protein alone, they're not very well. The representation is not as an immunologist would expect, basically. However, if we integrate them using fiber autoencoder, you can see that we recover the cell types exactly as expected. And even the fine cell types here, you can see they're very well grouped together. So this is another application, very interesting application of fiber autoencoders that I'm doing with Chris from the Helmholtz Institute. And this is the same thing, but a higher level of integration. You can see that the cell types here are very well integrated. And finally, for the last project, so there are many different models for single cell that have been introduced lately. So SCGen, CDR, total BI, scan BI, there's a lot of them. They're usually all variational, and they usually address the same problems, integration and bare cell representation, and they all use the same activation function. So the idea here we had, and this is a work I'm doing with one of my students, Fatem Zahra, at the NGS also, is could we improve all these models by introducing new layer types? So we're just going to try to improve all the single cell models at the same time. And these are the representations, the results we have, still preliminary, but very encouraging for the SCGen model on PBMCs, blood cells. And you can see that here we have we have metrics about the integration of the cells and the retaining of the biological information. So both of them here, basically the higher the better. And these are the different models. So liquid ReLU is the standard SCGen, and then we're trying to improve of that. And you can see that especially for the green native attention, we're getting better results on both cases for integration and then also retaining the cell type information. Same thing for this metric. This is silhouette, also measuring the same thing. We are able to improve on the results of SCGen by introducing our new type of layer for the integration, also for retaining the single cell biological information. So I end my presentation is over. So thank you very much for listening, and if you have any question, please feel free to reach out.
Thank you so much, Dr. Tariq, for your presentation. It was very interesting to know about the single cell and sequences transformation and the AI applied to those. So we have here some few questions. We'll start with the first one. So we have a question about the relationship between bioinformatics and NLP. So are there some specific neural network architecture that are tailored and adapted for bioinformatic applications that differ from NLP field?
There's always an adaptation that you have to make. Transformers, obviously, because they are applied to everything at the moment, have been applied very successfully to biological sequences. Usually, what you have to do is you have to basically have a different tokenizer that's adapted to the biological task you're trying to solve, and also you need to have at least some biological knowledge to know what you're looking for. But usually, if you're working on sequences, NLP models would work very well.
Thank you for your answer. So which take us to the next question, which is how do you collaborate with domain experts and what are the difficulties? And do you need to have a background in bioinformatics to explore this field, biological sequences?
Yes. Yes, you need one. So my background, I started with a background in pure mathematics and computer science, and I had to learn that basically you cannot just jump into biology because it's just too complex. So the best way to do it, in my opinion, is to find a problem that is well defined enough so that you can understand it. For example, do it basically the way DeepMind does it. Find a problem with a clearly defined metric, for example, to start, and try to improve on that metric, and at the same time bridge with biology, so they can you can get some information about some knowledge about what are the biologically relevant things to look for. And so that would be the way I would suggest starting. I wouldn't say you need some bioinformatics knowledge, but you definitely need some biology knowledge.
Okay, that's interesting. So the bioinformatic background is necessary to jump in the biological sequences.
Because the bioinformatics background is easy to get if you're already a computer scientist.
So within the same theme in our play models. So here we have a question about the tokenizers. Are they the same? Do we tokenize the mRNA as text, or they differ?
mRNA is basically mRNA and DNA is the same thing. It's read in codons. However, you can tokenize it different ways. You can tokenize it in codons. You can tokenize it in longer sequences. For example, in patterns, if you're looking for something very specific. For example, where other mRNAs would latch, for example, would fit on the mRNA, because it's biology is very complicated. Like there are a lot of things playing with a lot of things. So you can have longer sequences depending on what you're trying to do. You can also tokenize it by nucleotides, basically by letters A, T, C, or G. That also could work for some applications. So there are different ways of tokenizing mRNA, and they usually the easiest way is either by nucleotides or by codons. However, depending on what you're working on, you might have to tokenize it in more complex ways.
Thank you for your answer. So we still have 2 questions, then we will move out to the breakout room. So we have here a question about will AI help us accelerating vaccine trials as we saw in COVID-19 pandemic, or do we need other procedures to approve vaccines, such as to check biases in data training or other things?
I think we need more AI in that respect, especially for the safety data, because what we did, for example, the cocktail that I've presented is to create. So we run a neural network to predict for the best markers at the surface of the cells, but we also, so this is already a good head start. So it tells you what your vaccine should basically what should be presented at the surface of the cells, and what the targets of your vaccine should be. So this is already good. However, what we did next is we took the sequences and we tried to, and we removed all the sequences that were present in the human genome and also the mice genome. And this is to prevent, for example, autoimmune reactions or things like that. So what I think we could do with AI is basically we can use it to accelerate the research, but we can also use it to ensure that the safety of the product is higher. So we have less clinical trials to run. So this is also a way to accelerate the research. Finally, there's also people trying to build like virtual screening or things like that. These also are ways AI could accelerate the development of therapeutics.
Thank you. So this is impressive, and we are looking forward to seeing what AI can do in bioinformatics and helping in designing vaccines and molecules. So we will end with this question from a participant. He is asking about the explainable AI technique, LIME, that you presented. He's saying that LIME is known to be unstable to small perturbations. Have you tried other explainable techniques?
Yes, we've tried others, but LIME worked in our case. So for people who don't know how LIME works, basically to remove or replace certain tokens with other tokens and monitor basically how that affects the prediction. And in our case, with the length of the sequences we had, it worked really well. So we didn't have any problem with LIME. There are other ways you could use, for example, let's say that I'm a big fan of opening the network and looking at the features. This is not something that we did for the virus project because the networks were too complex and we had time constraints. But this is what we did for the Gamma project, and I think this is, in my opinion, this is the best way to go for neural network interpretation if you're doing biological research. Because if you can look at the filters, you can ask a biologist, does these filters make sense? and from that you can find hypotheses that you can test, and also like it really speeds up the research the research process. While methods like Lime, it's not as effective in my opinion to drive the research.
Thank you. So one last question before ending the Q&A sessions. So I will ask about Morocco. So what are applications that we can do in Morocco and how we can use those results in Morocco?
We can do everything in Morocco. What I would like to, I think, in my opinion, we should concentrate on problems that are faced, specific problems that are faced in Morocco and Africa in general, because these tend to be understudied and they don't get as much basically exposure as other problems. But I would say there's, at the moment, if you can get decent GPUs and decent infrastructure, there's absolutely no limit, because most of the data you would need is public. So the single cell data is public. You have millions of cells, because every single has to publish the data. So you have millions and millions of cells. The virus sequences that we work on come from the NCBI. It's also available. The peptides are also available. So there's a wealth of data that you can use and for which you can build models here in Morocco, anywhere in the world, as long as you have decent enough infrastructure. And I think today it's not hard to get that infrastructure.
Thank you, Dr. Tariq, for the presentation. That's it. Those are the questions that we had, and because the presentation was crystal clear, and I'm sure that the participants will ask more questions in the breakout sessions. So now we will move to the breakout sessions, but I will just stop the